All right. Good morning. So I'll give you a quick introduction as to what DeepLabCut does. I think probably most of you might have already heard of it, so it'll be quite quick. So as you know, animal behavior is very rich and complex. So even a little mouse is capable of some, I would say, surprising feats. So this mouse is jumping over here to get the little treats that are on this hanging cup, and he also has to navigate these obstacles that some carpenter made for him. And he lives in a barn, and it's kind of cool, in my opinion. So if even a mouse is capable of something like this, and we as neuroscientists have the ultimate goal of understanding what the brain does, then that means we have to make a good effort of trying to understand how these two things are related, particularly because the main output of the brain is behavior. So the main question now becomes then, well, how can we actually understand behavior? So here we have a bunch of dots that are moving, but even though they're just dots, I think it's pretty clear that it's two people walking between one another, right? And so the main idea that kind of surges from here is that, well, you can potentially just use dots at very specific joints or locations of a particular subject, and that might be already like a very nice low-dimensional representation of what the behavior that was being done, so to speak. And so with that, well, what is DeepLabCut? So it's a method for 2D and now also three-dimensional markerless post-estimation. Post-estimation referring to the labeling of these little key points of a particular subject. And so then the name markerless comes from the fact that you don't need these little markers on the subject. So in computer graphics or VFX, when you're doing motion capture, you often like have to put like markers on the actual subject. So that then, since these are very easily visible, then you can then transfer them on over to a 3D model of the person. So you don't need to do that with DeepLabCut. Instead, what you do with DeepLabCut is you use a very powerful neural network, in this case your brain, because that one can look at a single frame of a video and know that, well, yeah, these are the legs of the horse, the feet, the face, whatever, right? And so same with the mouse. Your brain is very good at already knowing, the little digits and are accurately labeling them. So the idea is that when you label your 50 to 200 frames, you then use these frames to retrain a deep neural network that then now will do exactly this job, but in for unseen video frames. And so, actually, in the back behind the scenes, what DeepLabCut is doing is using what's called transfer learning. So you have a pre-trained convolutional neural network that back in the day, like the resurgence of these models came up during the ImageNet competition, where you have something like 1.2 million images. Each of these images belongs to one of 1,000 different classes, and the competition was you have to make a model that does as best as it can in classifying each of these images. And so in 2012, it was the first time that a convolutional neural network was used in that competition, and it just blew the competition out of the water. And so what DeepLabCut is doing is it uses these already pre-trained networks to actually do its job. And the reason for that is because the trained network is actually a very good feature extractor. So it'll be able to know all the little edges, the textures, the shapes in general, and so you can take advantage of those that feature extractor to then use it to do the post estimation task. And the way you do that is as follows. So let's say this is your true video that you have. You only care about the paw, so you can like crop the location of the paw, and then you as the human, you go and label 50 to 200 of these frames, and then you pass it on to this network. And so this network is the pre-trained one, so you don't do anything to this. And what happens is you have these additional layers that get convolutional layers that get plunked on at the very end of the network. And essentially, what you're doing is you're only updating these layers to make it so that it can now label the frames that you gave it. And the idea is that once this thing is optimized, it actually works on frames that it has never seen before. And so with that, and so that's basically what DeepLabCut is doing. And so now what I will do is I will give you some examples of sort of what I did back in the day with this. So here I extracted the 1, 2, 3, 4, 5, 6, 7 points. And so I was able to then extract sort of like the pupil size and the pupil location on one, and exactly with exactly the same model, I can do that on a different eye as well, and it'll work well. And so these were supposed to tell me also the mouse blinks, so to speak, and so you can use this information then on your actual experiments. So now I will show you another thing that I did. So I had these mice that were trained on a task, so they were watching a monitor and they were moving a little wheel, and they had to do a visual discrimination task. So the task was the following: you have two stimuli. So you have this vertical in this direction and these oriented grading in this other direction, and the mouse had to rotate the wheel in the direction that would make this vertical. So if you're the mouse and this thing is what you're seeing, you rotate the wheel in this direction and this thing moves this way, and then you get, and what happens is you get water reward. So if the wheel moves, then you get the feedback, and if you do it correctly, you get water. If you do it incorrectly, you get punished. And at the same time, I am recording the eye as well as the body of the mouse. And actually also at the same time, I'm doing 2-photon imaging of the visual cortex of this mouse. So here we have like one hemisphere of the mouse. Here we have the visual areas. This is what this looks like at the wide field level. And very hard to see here, I'm sorry, there's the outline of different brain areas. And I kind of go in between, in this area, which is part of V1 and part of RL. And then onto here, we switch into 2-photon mode, and now in each of these little dots is actually a neuron. And like I said, at the same time, I was also recording the behavior of the mouse, so the pupil as well as the body itself. And using DeepLabCut, I extracted different features: the snout, the chin, which is sort of, if it moves, it means that the mouse is licking. I have the left, right paw, the pupil center, and now here I'm showing you these curves as a function of the experiment. So the chin moves, so these are probably all the times that the mouse got water. Here you have these like sharp movements here are actually saccades, so the mouse moved its eye, and then the pupil radius, it's kind of goes up and down actually with the task. And so then what I did is I trained a model. So basically what you kind of need to get out of here is that I go with, for each neuron, I then try to say, is there a way that I can combine my DeepLab extracted features to predict what the response of the neuron at time t was? And what I gave to the model was not just DeepLabCut, but I actually also gave it events of the task. So these like little blips are only when, for example, the stimulus comes on, or a different stimulus comes on, or the water comes out. And these orange ones are what I call continuous features that sort of have maybe the wheel movements, the pupil changes, among other things. And so here I train the model. So I have what's called a training set. So a training set is what is used to tune the weights of the model to get it to do its task. And then you have a set of the data that you haven't shown the model, and you present it, so that's called a test set. And when I do that, actually over 100 neurons, I cannot predict anything of what they're doing. Because even though over here, none of them have it, but that's because you can make a model be so good that it still will predict only this training data set, but that's not what we want. We want something that generalizes over into things that have actually not happened yet. And so over here, I have the test set data as like performance of the model. What I'm using to measure that is something called explained variance. So if you have a perfect model, you have 100% point, like 1.0 of explained variance that you can explain. But in this case, even in the test set, I have some cells that have maybe like 40% of explained variance, maybe some around 50 something percent explained variance. And so what this looks like is like this. So on the red, I have the prediction of the model, and in black, I have the actual neural data. And this is for the test set. So these are the predictions of the model for trials that it had never seen. And at least you can see here that it's like very nicely, to some extent, predicting some of the responses, but not all of them. And of course, I handpicked here. These are probably my 3 best cells. And then now I can go into a different kind of like analysis, which is called unique explained variance. And so what that is sometimes your features are correlated, right? So, and what I mean by that is, let's say that every time the mouse receives a water reward, that the pupil size increases. That means that even if I have the feature for when I give the little drop of water, it might actually be, the performance of the model might still actually get help from the pupil, even if I remove the water reward. Uh, and so that's what unique explained variance is trying to do. Is it's trying to find how much of the explained variance can only be possibly attributed to a specific feature. And so in this case, if you have an explained variance that's change in explained variance that's 100%, then that means that that cell was only being predicted by a single feature, if such a thing happened. But as you can see, my biggest drops might have been like 16% explained variance. And I think what's kind of interesting to me here is that you can find a lot of sort of heterogeneity in terms of what the cells seem to care about. So here P is the pupil changes, so a bunch of cells seem to be predicted very nicely by the pupillary changes. And some other cells are predicted nicely by C is for the chin, so that's when the mouse is licking. And for some reason on TE, that's when the mouse ignored the task and the task sort of ends. There's a bunch of cells that seem to care about that. And so yeah, this is a quick example of some of the things that you can do with once you extract features from DeepLabCut, you can maybe use them to try to understand the neural signature or how they relate to the brain activity. And that's pretty much it. That's the end.